Most research with liposomes rely on bulk techniques, intrinsically assuming all liposomes to be identical. By studying single liposomes, significant inhomogeneities can be observed. The single liposome technique presented here allows us to study hundreds of liposomes simultaneously and detect inhomogeneities in their size and composition.
The method can easily be adjusted to study other systems on a single liposome level, such as protein-membrane interactions. All you need is the compound you study to be fluorescently labeled. With this video, we provide researchers with a tool on how they can perform single liposome studies. We hope that it is clear that the assay can easily be modified to study a range of other scientific topics.
To begin, mix the prepared lipid stocks, 138 microliters of POPC and 120 microliters of cholesterol, in a fresh glass vial. Then, add 500 microliters each of two fluorescently labeled lipids and 50 microliters of DSPE-PEG biotin. Loosen the lid of the glass vial and snap freeze the vial in liquid nitrogen for 1 to 2 minutes. Lyophilize the frozen lipid mixture overnight in a freeze dryer. In the morning, add 1 milliliter of 200 millimolar D-sorbitol buffer to the dry lipids. Heat the mixture to 45 degrees Celsius and expose it to magnetic stirring for at least 1 hour. Then, freeze the lipid suspension by dipping the vial in liquid nitrogen and wait until the suspension is completely frozen. Next, dip the frozen suspension in a heating bath at 55 degrees Celsius until the mixture is completely thawed.